this is basically the cell theory historical timeline. What has been observed with time, how the cell theory came into evolution. So this is very important from the aspect of need as well, because most of the questions like who gave what point related to the cell theory and who first discovered the nucleus, who first discovered the cell are the questions which are basically asked. So, for example, firstly, the first compound microscope was invented in 1590. Later on, in 1665, Robert Hooke, as I told, was the first to visualize cells in the cork slice. Then in 1680, Anton van Leeuwenhoek observed living cells through the simple microscope. After that, in 1838, Matthias Kelden discovered plants are made up of cells. In the same timeline, around in 1839, Theodor Schwann discovered that animals are made up of cells. And then in 1855, Rudolf Virchow stated that the living cells, all the living cells come from the pre-existing cells, that is omnicellular, acellular. So this is just the summary of what I have told earlier about the cell theory. Any doubts regarding this? If anyone has any doubts or anyone want to ask any question? Okay. If there are none, I think we can move forward. So this is the overview of cell. So in this, we have taken two examples. Basically, two examples have been described. The first one is the onion cells, onion peel cells, which are the examples of plant cell. And the second one is the human cheek cells, which are the examples of animal cells. So onion cells, which is a typical plant cell, as I mentioned, that the plant cells contains a cell wall. So there are questions in which we are asked to differentiate between the two figures. And in those figures, in one of the figures, we are provided with the cell wall. And in the other figure, we are just provided with the cell membrane, that is a single membrane structure. So this is how we can differentiate between a plant cell and an animal cell in the theoretical or in the diagrammatic questions, which will be asked from us. So the onion cells, which are an example of a plant cell, has a distinct cell wall, and its boundary is just within its the cell, and within it is the cell membrane. While in case of the humans, we can say that since they are the animal cells, we just have an outer membrane as the delimiting structure of the cell. There is no cell wall which is present, and inside each of the cell, either be it a plant cell or an animal cell, there is a dense membrane bound structure which is called as the nucleus. So the nucleus is present inside the cell, and this nucleus contains the chromosome, which in turns contain the genetic material, which is the DNA. And in whole, this chromosome and this genetic material is called the nucleolus. So basically, all of the components are kind of similar between the plant cells and animal cells. First differentiating thing is just the presence of the cell. Now, cells are present in varied shapes and sizes. Their size differ greatly. Their shapes and activities differ greatly based on the activities. They acquire certain shape. Based on the activities, they have a certain size. For example, we can say the smallest cell. This is a very important thing, that the smallest cell, mycoplasma, which is also called PPLO, pleuropneumonia-like organisms, are the smallest cells, and they are only 0.3 micron in length. While bacteria, on the other hand, could be 3 to 5 micron. And if I talk about the animal cells, they can further be of larger sizes. So, the largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human blood red cells, human RBCs are about 7 micron in diameter, and nerve cells are some of the longest cells. As we can see that the nerve endings sometimes start from our head, and they may expand up to our toe. So nerve cells are some of the longest cells, and the smallest one is the PPLO, as I mentioned, which is around 0.3 micron in size. And the cells also vary greatly in their shape. They may be disc-like, they may be polygonal, they may be columnar, they may be cuboid. For example, you might have learned about the columnar epithelial cells, cuboidal epithelial cells. They may be thread-like or they may be even irregular. And the shape of the cell may vary with the function they perform. As I mentioned, that the muscles can be of various shape and size based on where they are present. For example, we have striated muscles. So they are of different type based on the, basically the shape is different based on the function which they are performing. So this is the example of the sizes and the shape of the cells which are present. So a typical eukaryotic cell, as I mentioned, for example, the RBCs in human, as I mentioned, is around 7 micron. So a typical eukaryotic cell can be 10 to 20 micron. While a bacteria is 1 to 2 micron, PPLO is around 0.1 micron, viruses are around 0.0 to 0.2 micron. While if I talk about the shape, then RBCs, red blood cells, are round or they may be biconcave in shape, as we can see in this figure. White blood cells are amoeboid and they acquire different shapes and structures. And since there are different types of white blood cells, most of the white blood cells differ from each other in shape. And so their shape is called as amoeboid. Long and narrow, that is the columnar shape, as I mentioned, or cuboidal shape I mentioned. So epithelial cells may be columnar or cuboidal. Nerve cells are branched and long. Elongated can be a tracheid, which is present in plants, and basophil cells are round or oval. Sorry. So there may be different shapes of the cells which are present. Now, based on the so this is the classification. This is till now what we have studied is what is a cell, why it is called as the basic unit of life, what are the different types of organisms based on the number of cells, that is unicellular organisms or the multicellular organisms, what is cell theory, who proposed cell theory, what are the different postulates which are given in the cell theory, and what are the different sizes and the shapes of the cells. The next is what are different types of cell. So basically, cells can be divided into two types. The first is the eukaryotic and the second is the prokaryotic. So what is the basis of classification of eukaryotic and prokaryotic cell? So cells that have a membrane-bound nuclei are called the eukaryotic. So this membrane-bound term is really important when we decide whether a cell is a eukaryotic or prokaryotic. So in case of eukaryotic cells, the nucleus is itself surrounded by a membrane. While in case of prokaryotic cells, nucleus is not surrounded by a membrane, as we can see in this figure. It is mentioned as naked DNA. Naked DNA means like the DNA is just present in the cytoplasm. It is not surrounded by any other membrane. While in case of eukaryotic cells, like in case of plant cells or in case of animal cells, which are a part of eukaryotic cells, we can see that the nucleus is present inside, like the nucleus is surrounded by a membrane, which is called as a nuclear membrane. So this is the basic classification of eukaryotic and differentiation between the eukaryotic and prokaryotic cell, that a eukaryotic cell do have a nuclear membrane, while prokaryotic cells lack a membrane-bound nucleus. While in both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. So cytoplasm is present in both. As I mentioned, cytoplasm is the main area where the cellular activities in both plants and animal cells takes place. Most of the chemical reactions which are important for the survival of the cell takes place in the cytoplasm itself. Most of the enzymes, most of the protein reactions, most of the enzymatic reactions takes place in the cytoplasm. So cytoplasm is the main area, main of the cellular activities in both the plants and the animal cells. Besides nucleus, eukaryotic cells have other membrane-bound distinct structures. For example, they have endoplasmic reticulum, which is again surrounded, which itself has a membrane, then Golgi complex, lysosomes, mitochondria, and microbodies. So here I want to like tell mention one thing, that the prokaryotic cells lack such membrane-bound organelles. So basically, in prokaryotes, we can say that beside nucleus, there no membrane-bound. Like nucleus, there is no other membrane-bound organelle. So in prokaryotes, there is no membrane-bound organelles which are present, while in case of eukaryotes, there are many membrane-bound organelles like nucleus, Golgi complex, endoplasmic reticulum, lysosomes, mitochondria, and microbodies are present. Ribosomes are present in both. As I mentioned, ribosomes are the universal organelle which are present in both prokaryotes and eukaryotes. So they are non-membrane bound organelles which are found in all the cells in both eukaryotes as well as prokaryotes. And within the eukaryotic cells, ribosomes are found not only in the cytoplasm but also on the surface of the endoplasmic reticulum. This we will discuss later in the later lectures when we will study animal cells or the plant cells in detail. But for now, it is important to know that ribosomes are not only present freely in the cytoplasm, but they are also membrane bound. So they are present on the membrane of the endoplasmic reticulum. And the presence of these ribosomes on the membrane of the endoplasmic reticulum give it a rough structure. That is why the endoplasmic reticulum on which the ribosomes are present are known as rough endoplasmic reticulum. While the endoplasmic reticulum on which no ribosome is present, they are known as smooth endoplasmic reticulum. So ribosomes may be present on the endoplasmic reticulum or they may be present in the cytoplasm. And within the two organelles, chloroplast in plants and mitochondria. So ribosomes may be present in the cytoplasm or they may be present on the surface of ER. If they are present on the surface of ER, then we called it as rough endoplasmic reticulum, and they may be present in the chloroplast in plants and mitochondria. Animal cells contain another non-membrane bound organelle, which is called centriole, and this helps in the cell division. So basically, in animal cells, two organelles like ribosomes and the centrioles are the ones that are non-membrane bound, which do not have membrane, and the rest of the organelles have the membrane. Similarly, while in case of prokaryotes, there are no membrane bound organelles. Here I want to tell a short trick, which is called as NAMAK. NAMAK stands for nucleus, mitochondria, and chloroplast. So this I will tell you later also, that nucleus, mitochondria, and chloroplast, that is NAMAK, is just the short form, or you can say a trick to remember that these three organelles are the ones which are double membrane bound. Like instead of a single membrane, they are surrounded by a double membrane. So what are these three organelles? These are like nucleus, mitochondria, and chloroplast. So I hope it is clear till here. What are eukaryotic cells and what are prokaryotic cells? So the next is some practice questions before proceeding further to the advanced topic. So what is the basic unit of life? Anyone who would like to answer in the chat? Or can unmute and answer? Okay, so we know that the basic unit of life is the cell. Who first explained that the new cells arise from the pre-existing cells? So the one who explained that the new cells arise from the pre-existing cell was Rudolf Virchow. And that was also the second postulate of our cell theory, that the new cells arise from the pre-existing cells, that is omnicellular acellular. What is the main area for the cellular activities in a cell? So as I mentioned in all the previous slides, that cytoplasm is the main arena for all the cellular activities that held in the cell, for all the enzymatic reactions, for most of the enzymatic reactions that held in the cell. So cytoplasm is the main arena where most of the cellular activities takes place. Now, which of the following is the smallest cell? as I mentioned, nerve cell was the one which was largest. Egg ostrich was also large. So largest human cell was nerve cell, while the largest cell was the egg of ostrich. Mycoplasma was the smallest cell, which was around 0.2 to 0.3 micron. And so the correct answer is the mycoplasma. Next is in both prokaryotic and eukaryotic cells, a semi-fluid matrix called dash occupies the volume of the cell. So the answer of this should be in both prokaryotes and eukaryotes, the semi-fluid matrix, which is called as the cytoplasm, occupies the volume of the cell, and it is the main arena for the activities of for all the cellular activities. The next question is, which of the following is correct? So the correct answer for this question is cytoplasm. The next is, which of the following is correct regarding the cell theory? Option 1, all living organisms are composed of cells and product of cells. Option 2, new cells arise from pre-existing cells. Option 3, living organisms are not composed of cells. And option 4, both 1 and 2. So basically, we know that the cell theory states that all living organisms are composed of cells and products of the cells. So option third is wrong, incorrect. And we also the second postulate, which states that the new cells arise from the pre-existing cells, that is omnis cellula is cellula. So the correct answer should be option number 4, both 1 and 2. Next question is, which of the following statement is not correct? So plant cells have a cell wall as the outer boundary, and just within it is the cell membrane. Second option is mycoplasma. The smallest cells are only 0.3 micron. Third option is nuclear envelope is present in prokaryotes. Fourth is the shape of the cell may vary with the function they perform. So we have been asked in this question, which of the following is not correct? So we know that the plant cells have a cell wall as their outer boundary, and within it, the cell membrane is present. So the option number 1 is correct. Second is, we know that the smallest organism is, the smallest cell is mycoplasma, which is around 0.3 micron. So option 2 is also correct. Option 3 states that nuclear envelope is present in prokaryotes. We know that the prokaryotes are the organisms in which the nucleus is not surrounded by any membrane. So therefore, option number 3, which states that nuclear envelope is present. Envelope, by envelope we mean membrane. So nuclear envelope is present in prokaryotes. So option number 3 is incorrect. So the which of the following? So since the question asks which of the following statement is not correct, so the correct answer is nuclear envelope is present in prokaryotes, because this is a wrong statement. Next is, cell wall is a unique structure of plant cells. Who concluded this? So we know that while studying the cell theory, it was Theodor Schwann who concluded that cell wall is a unique structure of plant cells. So the correct answer is option number 2, that is Theodor Schwann, who concluded that the cell wall is a unique structure of plant cells, and plant cells are composed of cell walls within which the cell membrane is present. The next question is, what do A, B, C, and D represent in this figure? So from this figure, firstly, we can have an idea that this figure is of a animal cell, since it only contains a single membrane. So therefore, since only the plasma membrane is present, therefore it is of a animal cell. So we can go one by one. So the outer membrane is definitely the plasma membrane. Next, the inside the cell, what is present? So inside the cell, we know that most of the cell is filled with the semi-fluid matrix, which is the cytoplasm. So this sky blue color here represents cytoplasm. So B is what? B is cytoplasm. Option C here pointed towards the nucleus, which is present inside the cytoplasm and which is itself a membrane-bound structure. So this C here represents the nucleus, and the option A, which is the membrane of the nucleus, represents the nuclear envelope or the nuclear membrane. So what are option A, B, C, and D? So A represents the nuclear envelope, B represents the cytoplasm, C represents the nucleus itself, and D represents the cell membrane or the plasma membrane. So the next question is, we have to match these following with their sizes. So we know that mycoplasma have a size range of 0.3 micron. Human erythrocytes have a size range of 7 micron. Typical bacteria are size range of 1 to 2 micron. And PPLOs, which are the smallest organisms or the smallest living organism, that is pleuropneumonia-like organisms, have a very small size of 0.1 micron. So basically, these are the correct answers of this question. So till now, is it clear, or any doubts in any of the questions? Okay. Hoping all the questions are clear. Moving forward to what is prokaryotic cells in pity. So the prokaryotic cells are represented by bacteria, blue-green algae, that is BGA, mycoplasma, and pleuropneumonia-like organisms. So as I mentioned earlier also, any doubts? Okay. So as I mentioned earlier also, in prokaryotic cells, no well-defined nucleus is present. So the most important characteristic of prokaryotic cell is no well-defined nucleus. So the genetic material is basically naked, not enveloped by any nuclear membrane. So nucleus is not well-defined. Why it is not well-defined? Because it does not have any membrane. And since there is no nuclear membrane which is present, genetic material is basically naked. By naked, I mean it is freely present in the cytoplasm. No organelles like the ones which are present in eukaryotes are found in the prokaryotic cells.
Ma'am, what is the difference between PPLO and mycoplasma?
Okay. So basically PPLO is like mycoplasma also, you can say it is a PPLO-like organism. So these organisms, PPLO is a different organism as compared to mycoplasma, but so mycoplasma are basically the free living organisms, and they are the these organisms basically lack the cell walls. So mycoplasma is when it is present in the, when it causes the infection and when it is present in the pleural fluid. So it was found that the so mycoplasma was basically found to be related to the infections related to the pleural fluid. So when it was found related to infection in the pleural fluid, it was named as the pleuropneumonia-like organism. So when the cell walls are, hello.
Which of the cell is the smallest cell, PPLO or mycoplasma?
So PPLO is the smallest organism, which is 0.1 micron, and
okay, ma'am.
Okay, any other doubts, or should I continue?
Shoot ahead, ma'am.
Okay. Sorry.
Yes, ma'am. Continue.
Okay. So basically, prokaryotes do not have well-defined nucleus, and they do not have any organelles which are present in the eukaryotes. So unlike eukaryotes, like they do not have any Golgi apparatus or any like endoplasmic reticulum and other organelles which are present in the eukaryotes that we will discuss later when we will talk about the eukaryotic cells. And as I mentioned, ribosome is present in both prokaryotic cells and eukaryotic cells. So, but prokaryotes have something very unique in the form of inclusions. So these are known as the inclusion bodies. And all prokaryotes have a cell wall surrounding the cell membrane except in the mycoplasma. So this is the basic structure of a prokaryotic cell in which a cell wall is present. Inside the cell wall, cell membrane is present. Then we have a circular chromosome which is present. And we have another different chromosomes which are different from the main genetic chromosome, which is known as the plasmid, which is present in the prokaryotic cells. These prokaryotic cells mostly have the flagella that helps in the movement of these prokaryotic cells, and cilia and flagellas are present on the cell surface, and they have the ribosome that help in the protein synthesis. So in addition to the genomic DNA, as I mentioned, the single circular DNA, which is present, many bacteria have many small circular DNAs outside the genomic DNAs. And these smaller DNAs are called the plasmids. And the plasmid DNA is important in the sense that it helps the bacteria to have the different ability. For example, the resistance to the antibiotics. We know that there are many bacteria which are getting resistant to the antibiotics. So this is because of the presence of the resistance gene against the antibiotics, which is present not in the main chromosome, but in the plasmid. So we can have a plasmid that contains a gene which makes the bacteria resistant to ampicillin, or we can have a plasmid which have a gene that makes the bacteria resistant to some other antibiotic like penicillin. So these plasmids confers the unique phenotypic characters to the bacteria based on the genes, or based on the based on the genes which are present on these plasmid DNA. So as I mentioned, one such character is resistance to antibiotics, which is very important, and it plays an important role
Ma'am,
yes.
what is plasmid, ma'am?
Sorry, what is plasmid?
Yes, ma'am. Tell me
Okay.
about plasmid.
Yes. So basically, what happens is, in case of prokaryotic cells, we have a main chromosome, which is a circular chromosome, right? So it contains the genetic information. But besides this, we have some other smaller DNAs which may or may not be present. Like these plasmids are not the necessary part of any prokaryotic cell, while the circular chromosome is the necessary part. So it is the main genetic material, but plasmids are not the main genetic material. They are present outside the genomic DNA. By saying so, I mean to say that they are like, as we can see in this diagram, you can see that there is a main circular chromosome, while there are other smaller DNA fragments, DNAs which are present. These are not the genomic DNA. These are the ones which are the plasmid DNA, and they confers the unique phenotypic characters. For example, as I said, that they helps in the attaining the antibiotic resistance. So plasmid in most of the cases, plasmid helps in the survival of the bacteria or in the harsh conditions. For example, it prevents the death of the bacteria in the presence of antibiotics. So for those bacteria in which the plasmids have that antibiotic resistance gene, they will be able to survive in the antibiotic, while those bacteria which do not have the plasmid plasmid against that antibiotic, or do not have the plasmid that codes for the antibiotic resistance gene, will not be able to survive in that condition. So basically, the plasmids are the ones that helps the bacteria to attain a certain unique phenotypic character. Is it clear?
Yes, ma'am. Yes, ma'am.
Okay. So plasmid DNA is also used to monitor bacterial transformation. So when we will in future, or maybe you might have studied in biotechnology, when we perform the transformation technique. So transformation is the when we want like a particular gene to be expressed in the bacteria. So this is a transformation technique which we generally use when we are performing any experiments in the laboratory. So when we want to express any foreign DNA, we perform the transformation. So what we do is we insert the foreign DNA into the bacteria. So this foreign DNA is inserted into the bacteria with the help of the plasmid. So this plasmid DNA is therefore also used to monitor the bacterial transformation with the foreign DNA. So this is about the plasmid. Um, Prokaryotes are generally smaller and multiply more rapidly than the eukaryotic cells. For example, we know that in case of E. coli, they just divide and replicate within the 18 minutes, while eukaryotic cells usually take a larger time to divide and multiply. They may vary greatly in shape and size. The four basic shapes of the bacteria are the bacillus. Bacillus means rod-like, as we can see in the figure. Spherical, that is coccus. Vibrio, which is the comma shaped, for example, Vibrio cholerae. And spirulum, which is the spiral shape. One more I have mentioned in the figure, which is the cocobacillus. So we can see in the figure that cocobacillus is a combination of coccus and bacillus. So it is not completely round. It is not completely rod shaped. It is a kind of oval shaped, and this is known as the cocobacillus. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. So the next is the prokaryotic cell envelope, and what are its modifications. So most of the prokaryotic cells, especially the bacterial cells, have a chemically complex cell envelope. Basically, the cell envelope consists of three structures. Outermost is made up of glycocalyx, followed by the cell wall, and then the plasma membrane. So in this figure, as I mentioned, in most of the prokaryotes, there are triple layer structure which is present. The innermost is the cell membrane, the next is the cell wall, and above that, a complex glycocalyx layer may be present. Although each layer of the envelope performs distinct function, but togetherly, what their function is to protect the bacteria. So together, they act as a single protective unit. Based on the composition of the cell wall, bacteria are divided into two classes. So they may be divided as gram positive or the gram negative based on the gram staining which they take. So gram positive, when we perform the gram staining over the bacteria, and if they appears to be blue, then they are called as gram positive. And if they appear to be pink or reddish type, they are said to be gram negative bacteria, as they are stained with the safranin. So glycocalyx differs in the composition and thickness among different bacteria, and it could be a loose sheath or slime-like in some, while it may be thick and tough, which is called as the capsule, in some other organisms. And the cell wall determines the shape of the cell and provides a strong structural support to the bacteria from bursting or collapsing. So cell wall as a whole is the, yes.
Ma'am, what is gram negative or gram positive?
So I will explain in more details about the gram positive and gram negative cell wall. So there are differences in the chemical composition. What are the composition of the cell wall? So most of the main composition of the cell wall is the peptidoglycan and lipopolysaccharides, right? So there is a difference in the amount of the peptidoglycan versus the amount of the polysaccharide, which is present in the gram positive versus the gram negative. And based on that, based on the amount of the lipopolysaccharide which is present in the gram positive and the amount of lipopolysaccharide which is present in the gram negative, these bacteria either acquire the crystal violet color, which is the, these bacteria either acquire the bluish purple color or the pink color, which is of the safranin. So if they acquire the blue color or purplish color, these are the gram positive bacteria. This is because they contain a very it will be better if I'll explain the gram staining in detail in the next class. I think that will make it more clear that why we are calling it as gram positive and gram negative. So when in the next class, I will explain about the structure of the cell wall of gram positive or the gram negative, or in general, the bacteria as a whole in detail. I think that time it will become more clear. Is it okay if I'll take it up in the next class, like what are gram positive and what are gram negative? for now, I think it is better if you understand like this, that in like the based on the staining procedure which was defined by Graham. So uh, uh, since it was based on the name of the scientist to give the staining procedure for the bacteria. So based on the cell wall composition of the bacteria either takes up the purple color or they takes up the pink color. So if they takes up the purple color, we classified them as gram positive, and if they takes up the pink color, we classify them as gram negative. But what is the actual principle behind why they are taking as purple color or pink color? For that, I think I need to mention I need a diagram that will help me to explain it in better details. So I'll take up it in the next class for sure. Is it okay?
Okay, thank you, ma'am.
Okay. Thank you. Glycocalyx differs in composition and thickness, as I mentioned, and the cell wall as a whole helps in preventing the bacteria from bursting and collapsing. So, thereby having a protective function. The next is the plasma membrane, which is semi-permeable in nature. So, it allows certain molecules to pass while it does not allow other molecules to enter, and interacts with the outside world. This membrane is similar structure to that of the eukaryotes. So cell membrane is similar in both prokaryotes and eukaryotes. Special membranous structures called mesosomes, which are the characters of prokaryotes, are formed by the extension of the plasma membrane into the cells. So these mesosomes basically, these extensions can be in the form of vesicles, tubules, or the lamellae, depending on the shape with which they are forming. And they help what is the function of mesosome. So basically mesosomes helps in the cell wall formation. They help in the DNA replication and distribution of the daughter cells. And they also help in respiration, secretion process to increase the surface area. And they also have the enzymatic content that helps in the like that helps in the cellular activities of the cell. In some prokaryotes like cyanobacteria are blue-green algae. There are other membranous extensions into the cytoplasm, which are called as chromatophores. As the name suggests, chroma is always related to the word wherever we are talking about the pigments. So chromatophores are the extensions which contains the pigments. So bacterial cells may or may not have these extensions. And if they have, like, for example, as I mentioned, in blue-green algae, so they have chromatophores that provide the color to the bacteria. Bacterial cells may be motile or non-motile. Motile means they have the ability to move. Non-motile means they do not have the ability to move. If motile, like, in order to move, they require something which helps them to propel and which helps them to move forward. So if motile, they have thin filamentous extension in the cell wall, which is called as the flagella. So this is the basic diagram, and bacteria show a range in the number and arrangement of flagella, and it mainly composed of three parts: filament, which is the longest part, hook, and the basal body. So the filament is the longest portion and extends from the cell surface to the outside. And hook is present inside, is embedded inside the cell wall, and the basal body is present completely in the cell wall and the cell membrane. Besides flagella, there are certain other organelles like there are certain filamentous structures which are present like villi and fimbriae, and these are also the surface structures that do not have the role in the motility. So what are the functions of the villi and fimbriae? So basically villi helps in the movement of the villi helps in the attachment of the bacteria, and fimbriae like these are made up of special proteins which are known as the pyrin proteins, and fimbriae are the small bristle-like fibers sprouting out of the cells. In some bacteria, they are like they also helps in the attachment, and in some cases these are also the root of the in some cases the pilus formation, that is the pilin helps in the this pili helps in the transformation or the transfer of the genetic material from the one bacteria to the another bacteria. through conjugation. So maybe later we will see what is conjugation. So through conjugation, this pili formation helps in the attachment. This pili helps in the attachment of two bacteria, and DNA gets transferred from one bacteria to another bacteria. So there are certain ways by which this DNA gets transferred. So one of them is the conjugation, and in that pili plays an important role. The next is the prokaryotic ribosomes and the inclusion bodies. So as I mentioned, in prokaryotes, ribosomes are present. In eukaryotes, ribosomes are present. But the composition and the subunit of the ribosomes differ in prokaryotes and eukaryotes. So in prokaryotes, ribosomes are associated with the plasma membrane of the cell, and they are about 15 to 20 nanometer in size. They are made up of two subunits. They have a larger subunit and they have a smaller subunit. So basically, it is composed of two subunits, which is 50S and 30S. This 50S and 30S, 50S is the larger subunit and 30S is the smaller subunit. These both together leads to the formation of prokaryotic 70S ribosome. Here S stands for the Swedberg unit or the sedimentation unit. Ribosomes are, as I mentioned, are the site of the protein synthesis. And in case of prokaryotes, several ribosomes may attach to a single mRNA, leading to several protein formations. And several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. So basically, as we can see in this figure, that this black color is the mRNA. So on the single mRNA, these blue colored structures, which are the ribosomes, are present. So many ribosomes are present on the single mRNA, leading to the formation of a structure which is known as the polyribosome, or in short, polysome. So the ribosomes of a polysome translate the mRNA into the proteins, leading to multiple protein formation at the same time. Now, what are inclusion bodies? So these are the reserve material in the prokaryotic cells that are stored in the cytoplasm in the form of the inclusion bodies. And again, these are not bounded by any membrane system and live freely in the cytoplasm. For example, they may be a phosphate granule, or they can be another pigmented granule like synophycean granules or glycogen granules. Gas vacuoles may also be formed in, and they are found to be present in some blue green algae and purple and green photosynthetic bacteria. So related to prokaryotes, there are certain questions which I have mentioned. So in prokaryotes, a specialized differentiated form of cell membrane structure is present, which is called as mesosome. So the options, like the answer is mentioned here, but the options may be mesosome, polysome, ribosome, or cell wall. So the membranous structure is the mesosome. Which of the following is not included in the cell wall of prokaryotes? Glycocalyx, we know, is present in the cell wall, cell envelope, sorry. Cell membrane is a part of cell envelope. Cytoplasm is not a part of cell envelope. It is present within the cell membrane. And it is the semi-fluid-like matrix, which is not a part of the cell envelope. So cell wall, cell membrane, and glycocalyx are the part of cell envelope. But option number 3, which is the cytoplasm, is not included in the cell envelope of the prokaryotes. Question 3 is mesosomes helps in, as we studied just now, that mesosomes helps in the cell wall formation. They helps in the respiration. They helps in the secretion processes in the bacteria. So all of these options are the correct answers. So mesosomes helps in cell wall formation, respiration, secretion process, or the division of the DNA in both the daughter cells. Prokaryotes have what type of ribosome? So 80S is present in eukaryotes. We will study later. 70S is the one which is present in the prokaryotes. This 70S is made up of two subunits, 30S and 50S. This we have to remember. Now, what is true for a prokaryotic cell? Like, they are smaller in size, they multiply much rapidly than eukaryotes. They greatly vary in shape and size, and all of these. So we studied just now that the prokaryotic cells are very small and they multiply very fast. They divide rapidly as compared to eukaryotes, and they have a variety of shape and size. So all of these are the correct answers. In prokaryotes, resistance to antibiotics is conferred by. So what are the things? What are the property? What is the the presence of what makes the prokaryotes resistant to antibiotics? So the option number 1 is plasmid DNA, option 2 is ribosome, option 3 is cytoplasm, and option 4 is genomic DNA. So as I mentioned, plasmid DNA is the one that contains the antibiotic resistance gene and therefore makes the prokaryote, respective prokaryote resistance to the antibiotic. So resistance to antibiotics is conferred by plasmid DNA in the prokaryotes. Now, next question is, which of the following is an inclusion body? Ribosomes, gas vacuoles, nucleus, and cell membrane. So the correct answer is gas vacuoles. Gas vacuoles are an example of inclusion bodies which are present in the certain photosynthetic bacteria. Which of the following is a pigment containing membranous extension into the cytoplasm? So options are plasmid, ribosome, chromatophores, and cytoplasm. So the correct answer is chromatophores. As I mentioned also that chroma means color. So chromato, like something related to chromato, is related to colors. So pigment are also something related to colors. So which of the following is a pigment containing membranous extension? So these are examples are chromatophores. Which of the following is a membranous extension into the cytoplasm? So we know that mesosomes are the membranous extension. So we know that the mesosomes are the membranous extensions which extend into the cytoplasm and have varied roles. So the correct answer should be option number 3, mesosomes. With this, I would like to end today's session. But in the next session, as many of you have the doubts about the gram-positive and gram-negative bacteria, I'll take up that topic in detail. So I'll explain what is gram-positive, why it is called gram-positive, and what are gram-negative and why they are called so. So in the next class, we'll study that gram-positive, gram-negative thing in detail. Also, if you people have any doubts, you are free to ask questions. Even if you want to ask it today, you may ask it now, or if you want to go back and read and want to ask questions in the next class, you are welcome to ask questions in the next class. And if there are no
Thank you,
questions,
ma'am.
should I end the session?
No, ma'am. Thank you, ma'am.
Okay, thank you.